The journey from an idea to a breakthrough is where science lives and breathes. Welcome to What's the Formula? Our series where leaders across science and medicine share insights about the formula for success in areas that will help us forge scientific breakthroughs. I'm Jane Wu, and we're here today with a few of our scientific leaders in the field of genetics and genomics. Sequencing the first human genome was considered one of the greatest feats in science and medicine. And now, over 20 years later, scientists have sequenced the most complete human genome yet. While we may have the blueprint for what makes us human, interpreting it is a whole other challenge. The field has yet to fully unlock the knowledge within to help answer our current and future questions about disease. And that's what we plan to talk about on today's edition of What's the Formula? Joining us are three leading DNA experts from AbbVie's Genomics Research Center. Mark Rappel, a statistical geneticist with a doctorate in biostatistics. He currently serves as a principal research scientist in bioinformatics at AbbVie. Bridget Riley-Gillis, a clinical molecular geneticist by training. She currently serves as a principal research scientist in human genetics at AbbVie. And finally, Nizar Smawi, a physician by training with deep experience in rare genetic disorders. He currently serves as a senior director in human genetics at AbbVie. Before today's event, we asked you to send in your questions about today's topic for our experts. Later in today's program, we'll feature your questions. Welcome, and thank you all for being here. DNA is the basis of life, containing the instructions for everything our cells do. Studying the human genome and all the DNA in our cells allows us to explore fundamental questions about ourselves and our health, like how many genes do we have, and what happens when we become ill. Genomics is swiftly moving to the forefront of medicine, transforming healthcare by making it more targeted, personalized, and proactive. It's an exciting time for the field right now. With new advances in technology, scientists can do things that wouldn't have been possible just a few years ago, like sequencing the last 8% of the human genome. To kick off today's discussion, I want to ask each of you to comment on the last 8% of the human genome. For many of us, we thought the human genome was already sequenced 20 years ago. So can you explain what all this buzz is about? So Nizar, I want to start with you.
So it's all started almost 20 years ago with the Human Genome projects, where there was this partnership between private and public. They built up this, what we call the blueprint, and what we called also a reference sequence. That was the first reference sequence. Since then, there was this huge and tremendous expansion of discovery related not only to genes for rare disease, but also for complex disease, such as rheumatoid arthritis or hypertension. And then, since then, the potential of new discovery for new treatment based on those new genes has increased tremendously. But in the meantime, we have this tons of trillions of data, genetic data that's being generated. Fortunately, we have Mark here, who is leading a team of computational and geneticists and bioinformatician, and to make sense of this tons of genetic data. And we have Bridget here as well to really try to understand the biology of these new gene and discovery. I would like to add that we thought that we had the final blueprint 20 years ago, but in fact, apparently we don't. And hopefully now we have the true one, that we are discovering this new region that hopefully we will find new genes. Hopefully we will be able to find even new therapy based on those new discoveries.
Great, thank you. Bridget, let's go to you.
Yeah, I think this is a really important milestone for the field of genetics and human genetics specifically. It's important to realize that the last 8% of the genome that was sequenced were really difficult regions to sequence through with the existing technologies at the time. There's large spans of repetitive DNA that technology just couldn't handle. And with the advent over the past 5 to 10 years of new technology and the accompanying computational approaches to analyze that type of data, it really allowed the scientists to complete that final 8% and allows us to now have a more complete assembly of the human genome. And it's important because we know that these complex regions are biologically important and will, as they reported in the paper, there's new genes and new transcripts that weren't previously known in previous assemblies of the genome. And I think it'll have, looking forward, a big impact on multiple areas of the genomics field. Personalized medicine, are there variants that are disease-causing in these regions, that Nizar alluded to, that could be important for personalized health? Tools and technologies that use the new assembly are going to really benefit, such as CRISPR genome editing. They have a more complete map for when they design guides that will target different regions of the genome. And then looking forward, I think this lays the groundwork to sequence more humans. So really represent the global population that we know should be more representative to represent everybody from across the globe and will have an impact on human health.
I mean, without question, it's very exciting right now. So thank you both for sharing your perspective. Mark, how does having a more complete human genome advance the research that you're doing?
Sure. So I think Bridget did a great job of describing, Nizar too, sort of what this means in terms of human health and for the human genome. I get really excited about how these technologies can be applied in comparative genomics. So high accuracy, long read sequencing, computationally feasible graph genomes, they really mean that people who are working in non-model organisms are going to have some resources that they can use to help build where they don't have a reference. So in particular, in the GRC, we have a collaboration with AbbVie's commercial organization, where they're using these hybrid cell lines to help produce our biologic drugs. And what this will, these hybrid cell lines don't have great reference genomes right now. And so some of these technologies make building de novo genomes really possible in some of these contexts, which could help us with discovering ways to improve the yield of the drugs that we're producing.
That's great. This is certainly a historic milestone for the field of genetics and genomics, as well as for medicine. And as Nizar and Bridget mentioned, you are using genomes as a tool to understand and unlock more clues about the underlying biology of disease. But how do we actually do this exactly? So Nizar, as a physician and a clinical geneticist and a drug developer, how are you using genomic data in your work to advance medicine?
So before joining the pharmaceutical industry, I was a medical director in a genetic testing company focused on genetic testing for rare disease. We know, and since the blueprint, since the first human genome project, whole genome sequencing and whole exome sequencing, we were able to identify a cause of these rare disease in 30 to 50% of the case based on the existing technology. We hope now with this new technology, we'll be able to even increase the yield to identify these rare disease. And we know for the drug discovery purpose, any genes that has been linked to a rare disease has 3 to 5 even more higher chance of success. So we will be more efficient in producing new drug and more efficacious and with less safety for these novel therapy. Basically.
Less safety concerns.
Less safety concern for these novel therapy. So this is a tremendous, huge, big change for us in terms of healthcare. And with AbbVie, we have this huge amount of data. We have access to the Million Genome, where we will be able to identify those we thought rare disease, but they're not as rare as we thought. Happy to talk about it later on. But that's the impact on this new genome will be extremely valuable for precision and healthcare in the near and even long term.
Yeah. Just so for those that are tuning in, when we say rare diseases, can you give us an example? Like, we're not necessarily talking about oncology, that's kind of widely known, but when you say rare diseases, what specific? Give us an example.
So there are, people think rare disease is where you have the syndromic disease, where you have some clinical features that, in addition of more severe end of spectrum, like developmental disorders. Rare disease can be as simple as familial hypercholesterolemia. There are cases where there are within family the chance of, if they have a mutation in the genes, the chance of inheriting these cases of familial hypercholesterolemia. That's a small, even like small clinical features. It is considered as a rare disease because its frequency in the general population is relatively rare. And like I mentioned earlier, this is not as rare as we think. And this is thanks to the whole genome and the whole exome sequencing data we know now across the biobank. They are much more frequent than we thought they are.
Great, that's super helpful. So thanks, Nizar. It's really interesting to hear how genomics impacts development decisions towards more personalized medicines. And at AbbVie, like you mentioned, we have 1 million genomes that we are using for research. So Bridget, how are you using this tremendous resource to advance our understanding of genetic components of disease?
Yeah, so AbbVie has spent a lot of time to build collaborations and join large consortia that are doing population-scale genetics research. So allowing us to have access to, these, quote, million genomes that are coming from resources such as UK Biobank that Nizar mentioned and FenGen, as well as harnessing data that we have from our internal clinical trials. And the power of this data is not just the genetics or the genomics that we have, but the paired information about the clinical health of those patients. So the phenotypic, deep phenotypic data is what we would call it. And that gives us the ability to ask certain questions and analyze this data in certain ways. One way is genome-wide association studies. And those types of analyses, we look for regions of the genome that associate with groups of individuals with a specific disease. And really the goal there is then to look in those regions of the genome, look at the variants that are associating with that disease, and then hopefully map those variants to a gene in that region. And then we try to take it from there and look, does that gene make sense? Can it be a drug target for these patients and to improve their health? And we know, as Nizar already mentioned, that publications from our colleagues and others that drug targets that have human genetic evidence supporting them are more likely to be successful in the drug development pipeline and in the clinical trials. So we're just trying to rethink, can we use human data, here specifically human genetics data, earlier in the process to improve the chance that our, the targets that we're working on will be successful and make it to our patients, and hopefully faster as well.
That's great.
Sorry, and to follow up on that, in the GRC also, we have Bridget's and Nizar's team and my team that are looking at this early on from the biobank perspective and the public research. But then within the GRC, we're integrated with lab teams, wet lab teams, like the functional genomics team, that can then take our hypotheses into cell lines, into CRISPR screens, to really try to push and see what we can do outside the public data that we have access to.
Yeah, which is, I think, one of the strengths that just right now Mark mentioned within the GRC. The GRC, the Genome Research Center.
Thank you.
That's right.
Thank you. Yes.
Thank you for that. That's
which
great.
is our acronym, basically.
Yeah.
We have 6 teams, that we start from the Genome Technology Group, where all the lab work, we have extensive technology, like the brand new technology, such as we're talking about. But also we have the functional genomics, understanding the mechanism of these genetic variants. And then we have the genome biology as well to work on the biology of the disease. And we have the human genetics and the genomics medicine, and that we all work as a team to really sift through this huge amount of data, basically.
That's great. Thank you. Thank you guys for that. Earlier in our conversation, we discussed how genomics relies on advances in technology. So can you talk about how new technologies have enhanced our research efforts at AbbVie? What are we doing differently now that we couldn't do years ago? So, let's start with you, Mark. What's your perspective on this?
Sure. So a lot of what we can do now that we couldn't do before has to do with scale, the speed at which we can do things and the ease with which we can do things. So 10 years ago, you might have been able to do something, but you had to do it by scratch, and you probably had to use all the compute that you could get your hands on to do it. And now we have much larger, more powerful computers, and you have standardized libraries and tool sets and file formats so that when you want to do something, it's just a much faster process so that you can do 10 studies instead of the time that it would take you to do one study before. And that's really, when we start getting up to 1 million genomes, you need to be able to do things quickly if you're going to have any sort of answer to come out of it. Yeah.
So to add to Mark, so I remember historically, when we started doing whole genome sequencing, we used to store the data on those hard drive, portable drive. And it takes 2 days to copy from the machine for one genome. Think about it right now. We have 1 million genomes. It would have taken months to just transfer the data. I'm not even talking about looking at the data, just to transfer the data. And good, the technology evolved so much, which is great.
Bridget, do you have anything to add here?
Yeah, I think, at AbbVie and in our Genomics Research Center, we've invested in bringing a lot of these exciting technologies in-house, which really opens up the space of the questions that we can ask, the tools and capabilities that we have. Similar to the publication that we're talking about for the new reference genome, we have long read sequencing capabilities. We also have other exciting tools looking at single cells, genomics, and spatial transcriptomics. And I think these really allow us to ask questions that just weren't possible because the technology didn't exist, several years ago. And, with the long read sequencing, we have the Oxford nanopore technology in-house now, and we're able to look at these complex regions of the genome in new ways, such as one area of interest for me is structural variation and how that has an impact on human health. And I think all of these types of tools, and then the ability to actually analyze it is really exciting and is really pushing the field forward quickly and gives us the ability to ask those questions internally as well.
That's great. Thank you to all three of you for sharing that. It's amazing to see how much the technology in the field has evolved and how many new discoveries it has unlocked. Mark, you specialize in bioinformatics, which is the intersection of statistics, biology, and computer science, and really vital to our genomics and genetics research efforts. Where do you see the field in the next 10 years, and what role will it play in our next major discovery?
Sure. I think for each one of these new exciting sequencing technologies, Oxford nanopore and single-cell RNA sequencing, each one of those comes with a need for the methodology to understand what you're seeing in your data. And so bioinformatics is really essential for the software, the visualization, the tools that let you get from a hypothesis to an answer of the question that you're trying to answer. It'll continue to grow with all of these diverse sequencing-based and array-based and all of the different technologies alongside those. It's going to have to continue to get faster as we get more and more genomes. If it takes you 6 hours just to read through the file with 1 million genomes, it's really hard to do meaningful analysis. And so to continue to scale and get faster, I think is going to be essential. And then, just visualization tools. So a lot of the data that we're dealing with now is highly multidimensional. And so you have your genomics, and in a multi-omic space, we have gene transcripts on top of it. You might have methylation, proteomics, and protein measures. And so how do you make something that the human mind can visualize what the data is even showing you once you start to get to those levels? And so I think there's a lot of room for expansion and improvement in those methods as well.
So just help me understand. So when you say data visualization, are we talking about like 3D, 4D? What does it actually look like for somebody who may not know what proteomics and all these other words may be? That's the issue, is that there are so many different ways to visualize it right now, and they are appropriate in different contexts. You can think about in genomics, as we talk a lot, you can visualize sort of the structure of your data
in principal component space, which is this multi-dimensional, and how you, so it gets very complicated very quickly. And I guess if you have a good answer, a good solution, that's what we're always looking for. It's
so basically, and to add to Mark's comment is, as the DNA, and we talk about genetics, the DNA goes and processes, and finally we have the protein. There is a lot of things that happens in between. And what we have been doing more recently is really looking at each part separately. DNA by itself, and we are not looking at the product of the DNA, which we call the protein. Now the challenge is really to put them all on top of each other. And that's really, to visualize that and to really look at it, that's one of the major challenges that we are having right now. Think about millions of data. Already genetic data, we have millions. Adding more data, it's going to be even more complex. But we have Mark here.
Yeah.
That's right. Well,
he has
definitely.
the magic.
The field of bioinformatics is definitely sounds very complex, but also very interesting. So definitely one to watch in the coming years. So, early in our discussion, Nizar, you and actually all of you guys have mentioned, and referenced 1 million genomes as being part of the key differentiators that brought you to AbbVie's Genomics Research Center. I'm interested in hearing your perspectives on the GRC and what makes it stand out. What makes you want to come to work every day and, ultimately play with the 1 million genomes that we have.
First of all, I love my job.
Yeah.
I've been telling everyone otherwise. I come and really enjoy everyday job, and because I'm passionate of rare disease, that's also I found all the support. Not only to the team who are here, and it's really a bigger, much larger team, but also the leadership at AbbVie, either the Genome Research Center or even the higher leadership. We have all the tremendous support from our leadership. And like I said, enjoying and getting help from, Mark and his team, Bridget, and the genome biology, functional genomics. Look, we are able to start not only from the DNA, and once you identify the gene or what we call the variant or the change, we can even go further. We are able to try to identify treatment, novel treatment, even for rare disease that we consider those incurable diseases. Now we generate lots of data, whole genome sequencing data from our clinical trials. And we try to see how they affect the response. And now we are finding that maybe some of our treatment that, can be used to treat those rare diseases, which is amazing. These are debilitating diseases, but we never thought that we will be able to do such things. So it's really exciting time for genetics, even more working at AbbVie and producing that data.
Yeah. Bridget, let me go to you. What does it mean to be able to come to the work at the GRC every day, the AbbVie Genomics Research Center, having access to the data that we do, and what that means in your everyday as you're kind of doing your research that will ultimately help patients?
Yeah. So I echo what Nizar said, being excited to come to work and being passionate about what you do. I think, at AbbVie, we always are really focused on the patient, and that's really at the core of all of the research that we do. And so keeping that in mind is really helpful. And when we think about why we are doing what we're doing. And so I think that's a really important piece of being able to come to work at the GRC and AbbVie every day. And I think also AbbVie has created a great culture around science and being a scientist. We're encouraged to publish, into literature. We're encouraged to go attend meetings and present at meetings. We collaborate outside AbbVie with other pharma partners, with large consortia, with academia. And I think that's really exciting because we're able to kind of tap into what the broader community is doing and try to bring that back in and ultimately bring that back in to help our patients. So do the research that really can make a difference and an impact.
That's great. Nizar, you talked a little bit about, how you're working at the GRC and how that supports early discovery research. And you also talked a little bit about I think, Mark, you talked about working with a commercial organization. But as you work, within the GRC, how does your work support even late stage development?
So initially, when the GRC was built, it was essentially a lot of discovery and a little bit of clinical development. Right now, we use the genetic data and genomic data that we are generating not only to inform our pipeline, to really what we call the reverse translation, which means we try to identify patients that do not respond to a specific therapy or specific drug, and then from there, can we then identify novel therapy, novel drugs for these patients, and then go back to our pipeline? So that's one. We work with across all AbbVie divisions, HOR, commercial. We inform even future clinical development based on phase 2 clinical trial data. Can we even build specifically a strategy for future clinical trial for other drugs? And this is the interesting part. This is not only for oncology. We are doing it for many other therapeutic area, the immunology, where AbbVie is the leading expert in that therapeutic area, but neuro and skin aging, aesthetic, and so on. So we are involved. I think the GRC, to be honest, is one of the few departments who is involved with so many other departments across AbbVie. We are everywhere. When we collaborate with different group within AbbVie, like I said, HR or epidemiology or medical affairs or others.
Okay, great. Thanks, Nizar.
Yeah.
And from the computational side, what do you think, Mark? And your teamwork within the GRC construct, how does that actually work and look like on an everyday basis?
Sure. So as Nizar alluded to, in the GRC, we get to go everywhere and work on a lot of different types of projects. So it's hard to describe sort of what a standard project or it fits in, but there's a couple different flavors. And a common one, Bridget and I were talking about earlier, an example of this is where a collaborator from a therapeutic area or another group might be interested in a particular gene for some reason. For example, someone in immuno-oncology might be interested in a gene that's associated with both immune phenotypes and also oncology-related phenotypes. And so they would come to us, and then we could look at the biobanks and public resources that we have access to, some of our proprietary algorithms that let us score sort of the links between these. And then we can return a report to them, giving them, here's the range of genetic indications that this gene is linked to. And if you have a drug that targets it, here might be some of the things that you would consider. And this can help with prioritization of clinical trials or other things like that. And then our team also gets involved a lot in building out the tools that everyone can use when I talked about standardization earlier. So if someone wants to do a GWAS study, we try to make it as painlessly as possible by setting up the software tools or the pipeline that they might want to run and use to do that analysis. And then we're also there if they want to call us up and talk about what their results look like. So you have someone who's done it before that you can consult with. And an example of this, our internal, so we have a sort of a sequencing collaboration center, the Genomic Technologies group that runs all of our next generation sequencing and assays and experiments. And they switched the protocol that they were doing RNA sequencing. And there was, the results looked different when it was done. And so they worked with our team, with the computational genomics team, to build a new pipeline to process that data. And then we worked with all the stakeholders in therapeutic areas as they started to get these new data that didn't look like the old data that they were getting. They could call us up and we could talk through what it meant and how they should use it, and sort of, instead of having to make oncology learn how to do that again and immunology learn how to do that again, we were able to centralize it and share that knowledge across the company.
That's great. I mean, I just find it fascinating that you can pick a gene and then figure out how to just study that one gene. So the fact that we have the capabilities now and the technology to do that is just really, really exciting. So thank you for sharing that, Mark. So I'd love to, maybe go back to you, Bridget. What can you give me another example of like how you're working with our teams in kind of doing this research, and what does that mean for those who are kind of new to this field? Like how do we understand what we're doing in the field of genetics and genomics into the kind of the everyday of what we do?
Yeah, I think Mark and Nizar have already given a lot of examples about how we can work with the various teams. So oftentimes the teams that we work with are in the therapeutic areas. They have a lot of expertise in the disease and the disease biology, but maybe they know a little bit about the genetics and a little bit about the genomics. And they come to us, we set up some questions around a specific disease area. We can look into the genes that are already known to play a role in that area. And then we can really like work with them to set up the questions. What other experiments need to be run to further answer those questions? There's all sorts of diseases we work with. We work in the immune space. We have a lot of interest in atopic dermatitis as an area that we spend a lot of time on, and just working closely with that team, building that rapport with the team, so that we can become the trusted partners in that area, and we can work with them to build the team to really further the science, so that it's not just the disease biology and not just the genetics and genomics, it's combining it to really power those experiments and those questions.
Great.
Yeah, and to follow up on Bridget, so we work another example with the clinical teams that are out there, and we say, as an example, we take Parkinson's disease. Parkinson's disease, one of the symptoms are tremors. And there is an unmet need for treating tremor in Parkinson's disease. What we do is physician and biologist and computational, we go and back to our existing biobank, and we try to identify a phenotype, Parkinson's patient with or without tremor. And then, Mark will run his GWAS, genome-wide association analysis. The result showed up, and interesting, and tell you, recently we found those. Bridget will take that and then try to understand the biology, and then from there we link the gene to the variant, what we call to the specific gene, then hopefully to the new drug that treats tremor in Parkinson's disease. So that's kind of an example where we have all this collaboration from the start, from the clinical and until the GRC, until the drug.
That's great. I mean, I think I heard from all three of you this culture of collaboration, the ability to work on really cutting edge kind of science and technology, and even, the fact that even in your kind of day-to-day doing this, that you're still able to kind of publish research. Is there anything else from a culture perspective that you want to share? Why? I mean, you already talked about why you're so excited to come into the office every day, but what about the two of you?
I mean, I do want to emphasize that the culture it's a very nice place to work. It's very collegial. People eat lunch together. They talk together. They have a good time. There's childcare on site, which I love. My sons commute with me every day. That's been a huge part of having a good life, a good balance in these things. So, the exciting chance to work on exciting technology in a very friendly environment has been great.
Very cool.
Yeah, and I think as we've all mentioned, it's very collaborative. You have the chance, if you're not the expert in the space, it's very easy to reach out and connect with those people that are within the company that are the experts. You can learn from them. You can continue to learn, pursue, advance your scientific career by learning just new skill sets. So I think that's really exciting that we have the ability to continue to learn, to continue to advance our scientific career, which may, a lot of times when you're on the academic side, you don't, think about that when you move into industry, that you can continue to build a scientific career when you move into pharma and other spaces.
Yeah. I mean, so I'm curious, how does one kind of get into this field of genetics and genomics? I know, Nizar, you're a former practicing physician. Sounds like you may have been in academia, but maybe I can just ask each one of you, how did you get into this field? Mark, do you want to kick us off?
Sure. I mean, so as an undergrad, I studied math and evolutionary biology, and I kind of wanted to find a field that I could, merge those ideas together, and went to graduate school in biostatistics. And I would say, at AbbVie, there's people of all sort of in each of those experience levels. We have people with computer science and mathematics backgrounds out of undergrad. We have people with master's level degrees in computer science, in biostatistics, and other things. And then we also, when you complete your PhD, we also have roles at that seniority level. And so it takes all sorts of experience and background. And so obviously on my side of the house, it's mostly computer science, statistics that people are working with before they come to work for AbbVie. But I know that molecular biology, chemistry are all skill sets well represented as well.
Great. What about you, Bridget?
Yeah, I think what really piqued my interest in genetics was the cloning of Dolly that happened while I was in high school, and I had a high school teacher that was really invested in us understanding science and the power of science. So I went into college knowing that I wanted to do genetics, and so during my undergrad, just tried to get as much exposure as possible. Work in labs where they were doing all sorts of types of genetics on model systems in the human, and then pursuing my PhD degree in human genetics was something that I just kind of knew that I wanted to do. So I would say for people that are kind of exploring this area is just get as much exposure as possible. Yeah. Try to get into labs, try to do internships, work in the summer. Even if you're just doing kind of the basics in the laboratory or in working with computational, get exposure to all the different areas of genetics and genomics that's happening.
Great. What about you, Nizar?
So for me, it started really when I started med school. I was talking to some of my friends, and we were deciding what specialty you want to do. And I said, you know what, I'm going to do genetics. And some of others were doing ophthalmology and surgery and so on. I said, you know what, I'm going to do genetics, so you guys will be out of the job because I'm going to solve everything. When anyone is, at birth, we will know the map of everyone at birth, and we'll be able to avoid all these genetic or, either complex or rare diseases. And that's really my passion. And then from that, started, being the medical director, like I mentioned earlier, in a genetic testing company. But then I said, yes, we're finding the answer, the gene causing this rare disease. We need to find the treatment. We need to find the cure or alleviate some of the symptom of this. And that's really one of the reasons, I find myself now switching to the pharmaceutical industry. So hopefully in 20 years, 30 years from now, I'll put out of business my colleagues, who are doing the other specialties. So that's really, my
that's amazing.
Story. Yeah.
Thank you guys for sharing that. Before we wrap up today's event, I want to ask each of you, what is the formula for success in applying genomic and genetic data for scientific breakthroughs, and how do you envision how your field will evolve now that the mapping of the human genome is now complete, so we like to say. So Bridget, let's start with you.
Okay. I think we've said it many, many times today, a diverse team of scientists. I think that's really critical to be able to ask and answer some of these large questions. We need everybody to be coming in with their diverse backgrounds to help solve these really big problems. This isn't really looking to the future. I don't think it's a single scientist or a small group that's going to be able to tackle the really, really large questions that are out there. And so I think being ready to work in these large collaborative teams with a diverse set of scientists is really going to be a key to success for the future of genomics and making those impacts that we hope to make in the healthcare field.
Great. Nizar?
So for me, I would say is really, and I echo what just Bridget said, and also we need to have more private-public partnership. This is not only AbbVie, this is many other, and we are involved in many other private-public partnership. And the question is really not generating the data, is working with this amazing team to really understand and interpret that data. I think that's really, we need to put more effort to really understand and manage that amount of data and put those different layers, like Mark was saying earlier about the proteomics all what we call the fancy word omics, basically anything related to X. We have to put it in one place to understand better the disease and identify novel targets. That's, I think, the where we should be and we should be going, basically.
Great. Mark, you want to close this out?
Sure. I mean, people is the key answer in terms of a million genomes is just a full computer hard drive without the people that you need to access it and analyze it and use that data. And so, yeah, I think making sure you have a team that's there, has hard problems, is working together and sharing their successes is really where you're going to find lasting success.
That's great. So I want to just turn to the audience questions with a few last minutes. So I'm going to ask the first question here. Do you think we'll ever get to a point where sequencing your genome is a standard part of healthcare? So I don't know, Nizar, you want to take that one?
Yes. So we are starting and we are there. I mean, whole genome sequencing and whole exome sequencing is now being offered as standard care, as part of the standard care for essentially rare disease. Still there is some pushback, but the hope is, like we are doing for newborn screening. When a person is born, not only in the US, Europe, or anywhere in the world, they will be able to have their genome generated and then will be able to treat them accordingly. Now, genome is being offered as what we call a rapid genome for critically ill newborn patients to see if these are caused by genetic disease, so to understand and to help treat them better. So we are getting there. Hopefully, it will be more in the near term and long term.
Great.
And I'd like to add just real quick that I think part of the onus is on the genetics and genomics community to also educate on how to use this information. Not everybody has expertise in this, and we know that, and we need to do a better job at educating the public, other physicians on how best to use this data as it becomes available, because it's coming.
Great point. So I think we have time for one more question, and the question is, the use of AI and machine learning in genomics is really in the early stages. How are we currently using AI and ML methods, and where do you see this being used in the future? So I'm wondering, maybe Mark, you want to take that one?
Sure. So, I mean, I think machine learning and AI are important tools in our toolkit. We like to approach the problem and ask what is the right tool to answer the question that we have here. And in some cases, machine learning or AI are the right approaches. So we've used them in the context of prediction or clustering, where we have large amounts of training data, other things that we can use. I think a limitation or an area where you need to be careful is you have to have that training data set in order for it to make sense and to be generalizable outside of its original context. And so sometimes when we're talking about rare disease or other things where there isn't a big training set, you have to go to your other tools. So it's not a one-size-fits-all solution, but it's something that we make use of in the places and times where it makes sense.
That's great. Well, I think that's all the time we have. So thank you guys so much for joining us today. I think we can all agree that there's a lot of excitement for the field of genetics and genomics. In the coming years, we'll be focused on generating insights and finding connections between our genes and health to better treat disease. So thank you to our panelists for sharing your experiences, lessons learned, and thoughts on the future of genetics today, and to all of you for tuning in and adding to the conversation. We look forward to staying connected, so don't hesitate to reach out to me with any questions. So thank you again. Our journey to discover the formula for success in areas that will help forge scientific breakthroughs and innovations isn't over. So stay tuned for updates and our next episode of What's the Formula, where we continue the conversation. Thank you.